Right, and the title of this is Using Organoids to Model Intestinal Regeneration and Colon Cancer. And so by way of background, one past one, this is a histologic image of the normal colon. And just like Omer, I'm also a pathologist, which is a doctor who, and one aspect of our job is to look at histologic slides. And it involves the visual diagnosis of disease by taking little bits of tissue and looking at it under a microscope. And so the normal colon is part of our digestive system. It has a lot of interesting and important functions. And one of them is to resorb water from our digested food. That's critical for our survival. And one thing that's unique about it is that it's arranged in these units called crypts, which are test tube-like shaped. And you can see that on the left here in this Alcian blue periodic acid shift stain section. And you can see they have a linear arrangement with the stem cells on the bottom and the differentiated cells on the top. And that's how the tissue regenerates itself. Unfortunately, though, this tissue can give rise to cancer, specifically colon cancer. And colon cancer is still the second most common cause of cancer deaths in the United States, with an expected 53,000 even in 2020. And so colon cancer is actually something we can model in the lab. So here is a histologic image of a colonic tumor. And you can see on the left, there is the normal crypts that we just talked about, where they have the normal organization and the cells are respecting each other's borders, and they are undergoing the same normal process of stem cell renewal and differentiation. However, on the right, you can see that evolving into a tumor. So a tumor has arisen inside the normal tissue and is giving rise to this mass, which is composed of colon cancer cells. And we know for colon cancer, the most common cause is actually loss of function of a protein called adenomatous polyposis coli, or APC for short. And it's the most commonly mutated gene in colon cancer. And we can mimic colon cancer in mice in the laboratory, which we do in the Yilmaz lab, by deleting the APC gene, and we can specifically induce tumors where we want to. And for example, here you can see on the right, we specifically deleted the APC gene in a few of the stem cells, and it generated this tumor in the mouse. One very convenient aspect of the intestine is that we are able to dissociate the crypts and grow them outside the body of the animal or of a person as mini tissues or organoids. And so on the left, you can see an image of a recently or freshly isolated set of crypts that were liberated from the underlying tissue. And then what we do is we actually have to embed those crypts into a hydrogel, which is a jello-like material. It's very soft and friendly for the crypts. And they're able to grow in media that we maintain in the lab. So we put them into these well plates, as you can see on the right, in a droplet format for convenience, which you can see there. And what we eventually then do after the hydrogel or jello-like material has set is we place media on top of it to permit their growth. And so that turns into these images that you can see on the left and the right. So the crypts actually end up growing and ballooning up because they no longer remember which side they were because they're embedded into a hydrogel. So on the left is the normal colon crypts after they've been grown for a few days. On the right are the tumor crypts after they've been isolated and grown for the same amount of time. So on the left, you can see how the normal crypts actually have a very balloon-like and cyst-like architecture and shape. And on the right, you can see that the tumor ones, though they also have a cyst-like and spherical appearance, they have some misshapen and unusual features and sizes. And also, they actually grow at an increased density compared to the normal because the cells are much more clonogenic or have the capacity to renew themselves. One convenient aspect of growing the cells in culture is that we are able to modify them to be fluorescent. Because since they're ex vivo and outside the animal, we are able to genetically engineer them, either using CRISPR or Cas9 or Cree technologies. And so after we make them fluorescent, Here's an example of that after we've modified them to be fluorescent. The fluorescent markers allow us to rapidly test conditions which may differentially affect the normal compared to tumor organoids. So the normal cells have been engineered to express a green fluorescent protein, so they look green, and the tumor cells have been engineered to express a red fluorescent protein, so you can see them here in red. So on the left, we grew them in a condition with media that permits the tumor to grow, and actually the normal is not doing very well. However, interestingly, on the right, this media condition selects for the normal cells to grow and outcompete the tumor cells, which is obviously something we're very interested in at the Koch Institute trying to treat cancer. And this is how we actually generated the image that we submitted for the KI award. And so this is that final image that was selected and is on the first floor of the image gallery. And I'd like to acknowledge the people that made this possible, as well as the funding sources. So Omer Yilmaz, the lab PI, Dayao Zhang and Camelia Huang, who are the UROPs or undergraduate researchers on the project, the rest of the Yilmaz lab, as well as the JAX lab for their expertise. And of course, the KI Image Awards themselves and Erica and her team for putting it all together, as well as the specific core facilities and funding sources that without which this wouldn't have been possible.